So okay. So what actually started green
Oh.
is coming?
Okay. So let me.
Sorry?
Can I have an autograph?
What's an autograph?
I mean, your signature.
Autograph. Autograph. Yes, of course. Okay, actually, I will do this here very quickly.
Yeah.
Right here?
Yeah.
Niin nimenomaan. Ja nyt mennään hyvän näköisiin.
Exactly.
Thanks.
All right. Now let's just get ourselves. Sorry.
Pictorial overview of today's.
Yeah, that's good. Yeah. And please fill in the middle. So fill the seats in the middle. This will be totally crowded. Yeah, that's great. Yeah, sure. Yeah. Yes, that's good. Yes, exactly.
I need to also connect this. I don't have a USB because this will have a USB. Let's see. Yes, it does.
It does, yeah.
Good.
But the power.
Right. There must be some blank the screen for a moment. Perfect. Press them again.
Yeah, you press the.
So this.
Both of the blue buttons. Yeah.
What does that mean?
Show the picture.
Show the picture. So I press those and it will come out. Do you have one on my chest?
We don't have one on the chest. We only have.
Do you have these things over my ears?
Yeah.
Well, what do you do with this? I never liked it.
I don't like them either.
Right.
I don't know.
Why don't you test? Let me know. This guy definitely works.
And it's on now, so beware. That's good. It's nicer because then you can walk around. I know you like to walk around.
I do.
Yeah. That's right. Just as a quick note, Lecture Hall 2 is also available for seating. You won't see Eric in person, but you can see him on the big screen, or at least his slides, I believe.
Yeah.
And the talk will be, I think, just as high quality in the second room. So.
Yeah, and there's more seats along the side here for people who want to come down. And there's room in the front, and
You're very comfortable with sitting on the stairs from the Cold Spring Harbor.
yeah, exactly. I always watch talks from sitting on the stairs.
Great. Okay, it's for me a singular and great pleasure to introduce Eric Lander to you today, though of course if there's any one person in our field who doesn't require an introduction, it's of course Eric. Eric has been without question the most influential and impactful scientist of the genomic era. I think starting in the 1980s, and that's when I first began my research as an undergraduate student in his laboratory, and what good fortune that was for me. And starting in the 1980s, when Eric laid the mathematical foundations of gene mapping that we use to this day, and of the mathematical foundations of how we would come to sequence the Human Genome Project. And then through the 1990s, in which Eric himself actually developed into a laboratory scientist and built and led the sequencing of the Human Genome Project himself. And through that was the driver of the development of the field of genomics. And then now even to the more recent past, where he has been the visionary leader of what is truly a visionary institution, the Broad Institute, and dedicated to the translation of genetics and genomics and technology to having true medical impact in a way that our field has not seen in the past. Along the way, he's also been a leader in ensuring that the products of our research, the data, the tools, the methods, are always freely available to researchers around the world. And he's also been a leader in public policy discussions, not only in biology and medicine, but in science in general. So we're extremely fortunate, and I'm personally gratified to have Eric here today, and his boundless energy, his curiosity, and his passion for impacting medicine and science with this research is still, to me, this day, amazing, and I think will come across truly to you in this lecture. Tervetuloa, welcome, Eric.
Oh, thank you so much, Mark. There we go. Can I do that? One of them worked, but not the other. Ah, there we go. I got them both now. Well, we'll see what happens. Anyway, it is an enormous pleasure to be here in Finland. There is no better and no more important place to give a talk on human genetics because, as I will describe, and as many of you know, Finland is so central to the whole story of the understanding of human genetic disease. And for me, it was an incredibly inspirational place. My collaborations in Finland go back a very, very long time. So it's just enormously special to be here. And this morning to get to see again many of my very close colleagues and to meet a lot of amazing students. Having Finn here and having projects like the FinGen project, it's just so inspiring that Finland continues to be at the cutting edge of human genetics. And you know, for a country of what's the current population? 5.5 million, you know, the impact often feels like 100-fold greater than that. And so it's really pretty fantastic. So today, I want to talk about some things that are on my mind. I like thinking about the big picture, being able to see the entirety of a discipline. And I think of it in terms of periodic tables. So the periodic table in chemistry, the periodic table of the elements, was transformative because it took chemistry from being something that was infinite and unbounded and, oh, there'd always be some other bit of matter, to saying, no, there only were a finite number of types of elements. When Mendeleev laid out his table, there were still holes in it. It still took 20, 25 years to fill in many of those holes. But of course, it changed our perspective on that world. Similar things have occurred in biology. It wasn't that long ago, the early 1980s, when I think biology seemed like it was infinite and always would be infinite. And things like the Human Genome Project said, oh, the genome, it's big, but it's not infinite. We could know it. And then human genetic variation. It's big, but obviously new mutations do happen, but it's not infinite. We could know most of it. And I think now we're living through another revolution that has started just in the past several years with cells. After the Genome Project was done, people would say, oh, there's never going to be another Genome Project because there's nothing else in biology that's really finite like that. But that's not true. I mean, the cells are finite like that. There are only a certain number of cell types, and we're going to know them all because of another revolution that is going on right now in a human cell atlas. And so I just think in terms of these periodic tables that become the reference points for all of biology. And I think we're seeing a steady succession of these things. And if you ask me in the question answers afterwards, I'll tell you I think there are more still to come. But I want to talk about it. And in particular, I've divided my talk into two entirely separate talks. The first part of my talk will be on common disease genetics. That's something very close to my heart, and indeed has emerged out of collaborations with my colleagues, particularly in Finland, over the years. And what I want to turn to is the next phase. I think the genetics of common disease is right now on the cusp of a change. I think for the last several decades, we've had phase 1. And I think we're about to enter phase 2. And I want to describe it. And I particularly want to describe it because it's still not fully formed in my own mind. And so I'm trying to write about it and think about it, and sometimes giving talks about where I think it's going is a good way to force yourself to be clear about it. But I think it's just a very special moment. But of course, to talk about phase 2, I'm going to also talk about phase 1 to lay the foundation, because I know this is a mixed audience. There's some of the most distinguished geneticists in the world here, but there are also many people who don't necessarily practice human genetics day to day. So I'll talk about phase 1, and then I'll talk about phase 2. Now I want to switch entirely to talk about a project that I'm excited about that flows out of the second periodic table, and that's inferring the trajectories of cellular development from single-cell sequencing data. And it's work that was just accepted and will come out in Cell soon, and I wanted to describe a little bit about that too. So that's the plan, and let's dive in. Oh, just to indicate how amazing our world is today, the first talk will have one slide that depends on 6 trillion genotypes, and the second talk will have one slide that involves 6 billion gene expression measurements. And the idea that one can talk about such things in individual slides is really quite stunning these days. It's not like biology used to be. So, the first talk, the genetics of common disease, phase 2. Well, to talk about phase 2, like I said, I want to talk about phase 1 first. So, phase 1 starts with this guy, David Botstein, a brilliant yeast geneticist at MIT, who had used DNA polymorphisms, DNA sequence differences, in 1977 to map a trait in yeast. usually genetic mapping is done by find a visible marker and trace it in a fruit fly cross or a yeast cross. But I ran into a trait in 1977. He couldn't really map that way, and he came up with the idea of using simple genetic spelling differences to trace the inheritance of a trait in yeast. And then the next year realized, well, if you could do that in yeast, you could do it in humans, where you couldn't set up crosses like in a fruit fly, and you couldn't use genetic markers like white eyes or curly wings or something like that. And he recognized that this would be a general way to map traits in any organism, but most importantly in humans. And that you could map simple monogenic traits caused by rare mutations by tracing their inheritance in families. He proposed this idea in a paper in 1980 that was seminal. It didn't just say, oh, you might be able to do it. It laid out in detail the recipe for how you would build a genetic map of the human being, and it was really the first idea of a human genome project was inherent in that paper. And so David laid this out. To show that Parkinson's disease was on next year. And David, as soon as he heard, this was a chance meeting, but when he heard that I both was a mathematician and a geneticist, he immediately, like within 10 seconds, started asking, okay, fine, we can do this for Mendelian traits, but unfortunately this is no good for common complex diseases. And we're both from New York City. He's from the part called the Bronx, I'm from the part called Brooklyn, and we immediately fell to arguing, which is what people do in New York City. It's a form of discussion. And we started arguing about it. I said nonsense, or I think I probably said it more strongly than that. And we began arguing about whether or not you could apply these tools to dissect common diseases. We had so much fun arguing. We got together the next day and argued some more, and the next day and we argued some more, and out of it emerged some initial ideas. David was going to give a talk at Human Gene Mapping 5, the 5th Human Gene Mapping meeting, and he invited me to come along because, in fact, he was going to talk about some of this work we were doing together. And at that meeting, there was a very fateful conversation which took place with Albert de la Chapelle in the restaurant Capelli on the Esplanade. And it was the conference dinner. And I was sitting across from Albert, and David was there. And Albert was describing that which is well known to people here, which is that Finnish genetics is unusual. That there are some diseases at much higher frequency in Finland than elsewhere in Europe, and some at lower frequency. That the geographic distributions are anything but uniform. They show concentrations in different places. And that this was the clear sign of a bottleneck that was associated with the founding of the Finnish population some 2,000 years ago, with a bottleneck that we now think has an effective size of about maybe 100 chromosomes, give or take. And what occurred to me at that dinner was, well, if that was the case, then whatever chromosome had brought in this mutation into Finland, this red chromosome carrying that orange mutation there, well, it would still be the case after lots of genetic recombination that we would recognize the ancestral segment. Now, that at least it would be carrying the ancestral segment. So, this idea of using genetic maps, Botstein had been thinking about doing this just in families, but of course you could treat all of Finland as if it was one very big family. And if your map was really, really, really dense, you would still see those segments. So all you needed was a super duper dense genetic linkage map, and you would be able to look back into history and see these segments and see that people who had any particular trait or condition that was due to a rare Mendelian mutation had a segment in common. In fact, collaborating with Albert de la Chapelle and Jona Hestbacka, who I'm delighted to say is in the front row, we actually worked together to apply this to a very important disease here in Finland, a disorder called diastrophic dysplasia, and it behaved exactly as theory said it should. And that was a good thing. And then I also want to say that in the course of many discussions after that, I also got to collaborate with Leena Peltonen, another great Finnish geneticist, all thinking hard about the amazing opportunities here in Finland because of the unusual population structure and the great scientific community. So that was one question. It turns out you could apply things by not looking within a family with meioses, but thinking back about ancestral segments. Another question that was of great interest at the time was, could you map things that were not single gene disorders, but were polygenic, that were combinations of many things working together? And at the time, you couldn't really do that in humans, but I ended up collaborating with people in agricultural genetics and walked around, even though I'm from New York City, walked around a lot of cornfields and collaborated with people who studied tomatoes. And we showed that, again, using genetic maps, one could dissect polygenic traits. So if you put together the pieces, in theory, there was this nice method developed by David Botstein for rare monogenic mutations traced in families using relatively sparse genetic maps, and the idea that if you had really dense genetic maps, you didn't need the family tracing anymore. You could use whole populations to find segments, and maybe in principle you could do this if it wasn't monogenic but polygenic. So those were the ideas. But of course, to make anything practical, we had to confront the fact that the human genome was very big. It was 3 billion bases. It still is 3 billion bases, I note, but it's no longer very big, because 3 billion bases isn't big anymore. But back then, that was big. And you were looking for, a gene and then a single mutation. And so the whole world got together and sequenced the human genome. There was this first Botstein-inspired and Botstein-founded genetic map in 1987. And then this 13-year Human Genome Project involving groups around the world. We had our own group in Boston that was a major contributor to this, and it eventually got done. I leave out a lot of details in that, and in a different setting and with appropriate beer or wine could say much more. But I shall not, to simply say the Human Genome Project got done. And the real question is, what did it mean for this mapping of disease? Now that you had a human genome, could you really apply these methods? Now, they were done for cystic fibrosis and Huntington's, but without the maps, it was slow and expensive. So the history was this. For Mendelian disease, this worked beautifully well. Family-based tracing in the families with markers for Mendelian disorders worked very well. But for common diseases, it didn't really work. Efforts to try to map common diseases by family-based linkage analysis, things like SIB-pair analysis, produced almost nothing. Not completely nothing, but almost nothing. The handful of genes, and they didn't look like the rare, highly penetrant, strong alleles that caused most rare Mendelian diseases. When you found any, and you didn't find very many, but when you found them, they had modest effects, a 2-fold, 3-fold, 4-fold increase on risk. So clearly, you had to think differently about this. Common disease was not just a collection of lots of rare Mendelian diseases. So two very influential things happened around 2001. One, working with David Reich and thinking about what would you expect the alleles for common disease to be, it became clear that for a population that had undergone an exponential expansion like the human population, the allele frequency distribution still should look like the smaller population. It takes a very long time after an exponential population expansion for allele frequencies to shift, all of which is to say there is very good theoretical reason to believe that common diseases should be caused in many cases, probably most cases, by the action of common variants. So there was a strong theoretical idea. And therefore, maybe this idea of using ancestral segments, like in Finland, would work. Because even in very big populations, there would be common variants that lived on segments that had arisen in the population, and you could still detect them. So Then the question was, well, how big were these ancestral segments? Was it a little distance, a big distance? Was it practical? And here, Mark Daly's seminal work in really exploring for the first time in detail the haplotype structure of the genome showed us that there really was a clear haplotype structure, at least in one region on chromosome 5, that extended over significant differences and was actually very elegant. It had haplotype blocks and things like that. And then quickly following up on it, Stacy Gabriel demonstrated by looking at 30 more regions of the genome that this was indeed quite general. So we knew what the scope of the problem was, that we would need to be able to recognize ancestral segments of 30, 40 kilobases, something like that. And in Mark's paper, there's even the specific proposal that based on the work, we should build a haplotype map of the entire human genome. And within a few years, that was done, or at least launched within a few years. By 2003, a haplotype map project was launched. By 2007, it was largely completed, and we had an idea of the correlation structure across the whole genome. So, you know, it's an amazing thing. You have a very dense map, and again, I want to emphasize this because this now seems obvious, but it was truly crazy that we should say, in order to do this, we need to know all the genetic variation in the human population, or almost all of it, all the common genetic variation. We need to know the haplotype structure of the whole genome. We need ways to be able to genotype not one marker at a time, like was done in the late 1990s, but hundreds of thousands to millions of markers in tens of thousands of people. That was insane. And we needed rigorous analytical methods to make sense of all of that. And the answer was the scientific community came together and did it. I emphasize this because I think in phase 2, there are more projects like this that are going to have to get done to build an infrastructure. And we should not be scared about rolling up our sleeves to build infrastructure. Again, a tremendous amount of work went into this by many, many people, but we got to the point where we have virtually all common genetic variation in most major continental populations that have been surveyed well. On your phone you can look up, or take any site of heterozygosity in your genome, and the probability is well in excess of 99% that site is already known and is available on the web. We know the haplotype structure, and we can genotype with genotyping arrays up to millions of markers, and then infer statistically the rest. So we can infer almost all the other ones. It's kind of mind-boggling, but anyway. And then sequencing. Sequencing has changed radically. We spent like $3 billion on the first human genome, which is a lot of money. But the cost of sequencing has now fallen by 5 million-fold. I don't know what else in your life you have seen that's fallen by a cost of 5 million over the course of 15 years, but even Moore's law in computing runs a lot slower than that. So, I mean, just think 5 million-fold. If it cost 5 million-fold less to buy a house or something, or fly to the United States, it's like inconceivable that something fell 5 million-fold. But of course, it means that we can routinely sequence genomes, and I predict it will fall to $100. It will be $100 perhaps 5 years from now. I have pretty good reasons to think that's the case. And when you figure, somebody lives for, let's say, 100 years, it costs $100. The amortized cost is $1 a year to have your genome. It's just mind-blowing. All right, so anyway, I digress. What about mapping disease genes? Let's come back to that. So I told you that simple Mendelian disease genes, that worked pretty well. But what about these common diseases? Well, to summarize this critical period right around when these tools became available, I offer you this slide that shows that in the beginning years of the century, 2000, 2001, 2002, basically worldwide, there was about one discovery in the entire world of a gene affecting a common disease. But then in 2005, there were 4 discoveries. 2006, there were 8 discoveries. And that's because the tools were beginning to get better. And by 2007, a huge number of discoveries had arisen, because suddenly the maps, the haplotypes, the genotyping were all becoming doable. You would imagine people would be very happy, but that is not the way we are in science. When we see something, of course, we say, okay, sure, but. So the but was, yeah, well, that's great, but for any given disease, there's only a few loci that have been found. They're mostly in non-coding regions. They only have small effect sizes, and altogether they don't explain much of the heritability. This whole thing must be wrong. And there were debates. There were some who argued, oh, well, you know, it's really got to be rare variants, like in Mendelian disease. Of course, that had been tried and hadn't worked, but it didn't matter, because the pendulum swings back and forth in science. But of course, there's another possibility, which is that it's not rare variants, it's just your studies are grossly underpowered, and that's why you're not finding a lot. So a good example of that was work of colleagues, Mark Ben-Neil and others included, in schizophrenia, where initial genetic efforts in schizophrenia to look at 3,000 people with schizophrenia, 3,000 people without schizophrenia produced absolutely nothing. Not a single significant locus was found anywhere in the genome. And this is depressing. It was especially depressing because the NIH study sections who would review a next phase of this said, well, we're certainly not going to fund any more of this because it doesn't work. But of course, even though nothing reached the incredibly strict statistical threshold, 5 times 10 to the minus 6th or whatever, you can still tell that something's going on. Because if you look at the p-values were not uniformly distributed between 0 and 1, which is what you'd see if there was nothing there. They were bunched up. They were basically trying to tell you, we're trying to be significant, give us a bigger sample size. And sure enough, with 20,000 samples, there were 5 significant loci. With 50,000 samples, there were 62 significant loci. With 110,000 samples, there were 108 significant loci. And with 163,000 samples, 245 absolutely robust, statistically significant loci associated with schizophrenia. This is not unique to schizophrenia. There are more than 100,000 associations between a trait or disease and a locus in the human genome. And you know, just for ones that have been worked on in detail, type 2 diabetes, inflammatory bowel disease, which Mark has done landmark work in, coronary artery disease, schizophrenia, there's several hundred loci that are known today. Moreover, we now know, of course, from the story I've told you, that there must be many more still below significance. And Peter Vischer in Australia worked out an elegant way to figure out how much they're contributing even before you know which ones they are. So they're below significance, but the bulk contribution was clear, and Vischer was able to figure, and one can roughly say, depending on the trait, that maybe half of the heritability of these traits is clearly due to common genetic variations that you can detect. Very likely, in my opinion, that interactions contribute more, but you can show theoretically that it's incredibly hard to find them using existing techniques. So you can't see them, but again, power would say you have no right to see them. So we don't know how much more may be due to interactions amongst those things. So has this given us insight into disease? Answer, yes, kind of nicely. Of these 100,000 loci, I'll just pick a couple of diseases. I'm not going to go into detail on them, but just to describe, in inflammatory bowel disease, where, as I say, you know, Mark and Ron Xavier have done just landmark work, and there are 225 loci. They've identified something like 10 new pathways, including protective alleles, protective loss of function alleles, that is knocking down the gene, has the effect of protecting you from disease, which, by the way, suggests that an inhibitor of the protein will be potentially protective in processes like autophagy and TGF-beta signaling. Autophagy is a great example of the power of genetics because if you did a Google search on the number of scientific papers mentioning the word inflammatory bowel disease and autophagy, the number of such papers before this genetic finding was 0. You can actually calculate it. It had not been thought about as a pathway, and now is recognized as an incredibly important pathway. Heart disease. Sek Kathreson has done beautiful work in early onset cardiovascular disease and working together with an international consortium. It turns out that although we all know that lipids matter a lot for cardiovascular disease, when you ask in an unbiased fashion which loci matter, well, only 20% of the loci have any effect on lipids. 80% don't. So it tells you there are a lot of loci that don't act through lipids, and that's really interesting. In addition, it tells you you can learn things by processes called Mendelian randomization. Just looking at what did people inherit? Alleles that raise their triglycerides a little bit, their LDL a little bit, their HDL a little bit. You can ask questions like, who's protective versus who's a correlate? And in a beautiful study exploiting the genetics, Sek showed that those people, so you all know HDL is good cholesterol. You want to have high HDL, and drug companies have spent billions of dollars trying to create drugs to raise your HDL. Well, Sek said, some of you inherited alleles that raised your HDL. Some of you inherited alleles that lowered your HDL. Did you get any benefit? Answer, no. Absolutely no correlation with protection amongst people who inherit raising or lowering alleles on average. And, oh, by the way, the drug companies that spent billions of dollars, the drugs failed because the genetics probably could have told you that in fact, the reason HDL is a good cholesterol is because it's inversely correlated with triglycerides, and it's triglyceride levels that really matter, not the HDL. It's a very good epidemiological predictor, but it's not the causal factor. Schizophrenia has pointed to synaptic pruning. The biggest of those signals is a locus that encodes complement factor 4 involved in the pruning of synapses. And it, as well as other genes on the list, suggests synaptic pruning is an important pathway. And work in body mass index, beautiful work of Melina Klausnitzer, and I'll return to that in a second, has identified particular pathways there. The problem is these 100,000 associations are mostly non-coding, but very few have been wrestled down to here's the variant that affects the gene that works in this cell type, affecting this pathway and producing this physiology. In fact, even getting from the non-coding variant to the gene is slow and painful. There are beautiful stories I've cited here. Each is heroic in its own way in the work that was done to truly wrestle these things down to the ground when you didn't know in advance, that this was a gene already involved in a process. We got to make this much easier. Now I'm going to digress for just 1 minute to describe a new one, which is from Melina Klausnitzer, and I've summarized what could be a very long talk on one slide. But it's beautiful work that she's readying for publication, and she and I have been working on this together. And I'll just give you this as an example of what one can learn, and also that it's still way too much work. So what Melina was interested in is the observation that type 2 diabetes patients often have high bone density. High glycemia is epidemiologically correlated with high bone density. Incidentally, though, also with higher fracture rate, which is seemingly paradoxical, but I won't get into that. So she looked for regions of the genome that showed some pleiotropic association with both high glucose, high glycemic traits, and high bone density. And because of these amazing databases that have been assembled in different studies, disease studies and biobanks and things, you can do that. And so she found that there was a particular haplotype that showed evidence of both being related to those two traits. And it was in an intron of ADCY5, an adenyl cyclase gene. So here's the genetic findings in a nutshell that showed how she's pinned the thing down. The haplotypes, the two haplotypes of that locus, are associated with differences in chromatin accessibility in vivo and also enhancer activity in vitro. If you take out the haplotypes, you can show in an enhancer assay, and if you look in patient tissues, you can show in a chromatin accessibility assay that the haplotypes show differences in adipocytes and osteoblasts derived from mesenchymal stem cells. In addition, the two haplotypes are associated with differential expression of ADCY5, that gene, and no other genes in the neighborhood. So that's likely the target gene. If you then look at the 13 possible variants, the SNPs in the region, and do certain types of computational analysis, either by evolutionary conservation or by a kind of deep learning algorithm to ask which ones are more likely to be play some important biological role in changing something, both evolution and deep learning agree that this SNP over here is the most important SNP. That SNP turns out to disrupt the binding site for SREBP1, a protein that is involved in both cholesterol metabolism and lipid metabolism. And if you then try to knock down SREBP1, you show that it indeed affects this adenylate cyclase gene. You can show that the SNP and the gene expression are associated with lipid oxidation in adipocytes and with osteoblast differentiation. You can show that if you change the expression of this this adenylate cyclase gene, it itself alters the lipid oxidation in adipocytes and osteoblast differentiation. And then if you take mesenchymal stem cells and CRISPR edit in an isogenic fashion, the one base, you can reproduce the phenotype. So it's that SNP. It acts on ADCY5, which has effects on lipid oxidation and osteoblast differentiation. It's a beautiful story which is being written up at the moment, but I bring it up both because it's beautiful and because it's a lot of work. And we need to make this like much easier. So there's a lot of interest right now, and Mark and I and many others are talking about phase 2 of human genetics, from variance to function. How do we move much more rapidly down this path from variance to function? And I think we need to think in the same way we thought about phase 1 of human genetics, where we collected all the variation in the human population and all the haplotypes and built the technologies for genotyping. We've got to ask, what do we have to do to just make this easy? So I've been experimenting with different ways to draw this out, and I'm not going to walk you through all of the details of it, but there are many steps along the way that we should think about doing. We don't have to think about doing this sequentially. It doesn't have to be to go from samples to variants to genes to cell types to processes to functions. We can sometimes leapfrog from one to the other and back again. But ways of using, for example, these genome-wide association studies, get more loci for any individual disease, fine map if you have larger sample sizes, but also annotate all the loci based on the other traits that they also affect, what we would call a phenome-wide association study. The more traits we have, we can cross-annotate all of these things. We need ways to build regulatory links between any enhancer and any promoter. Indeed, we just need a lookup table for every enhancer in the genome. We need to know what it's affecting in every tissue. We need to be able to look at rare genetic variants. I've been focusing on common variants, but rare variants matter. They're just not that common, of course. They don't explain that much, but they could be very informative. We need to find them. And then there are these ways to take these GWAS signals with 6 million genetic variants and learn from them. Even though you don't know which ones are significant, you can correlate them with things. You can see what they're enriched with. And all of these just depend on generic databases that we could build as a field. Below the red line, there are also disease-specific experiments that you will do, and you should do them, and they will require specific cell type models or organoids or something. So I'll talk just about a few of these to give a sense of what the state of the art is, but there's lots more to do. Rare variants. So I've said so much about common variants. Rare variants, they're important, but people, in the faddish way that it is, said, okay, we should go find rare variants. Let's go sequence 100 people. You don't see anything when you sequence 100 people. They say, let's sequence 1,000 people. And so people did that, and they were very depressed that they didn't find anything. But of course, there's a power question. Like we found with the common genetic variants, there is a natural power. How many samples do you need to have any right to expect to find something? And so if you do the power calculation, the answer is you shouldn't be talking to me unless you have 25,000 cases that have been sequenced, because we know roughly what the background rate of loss of function mutations are in a gene, and the background rates are on the order of a couple per 10,000 chromosomes. So if you haven't got things on the order of many 10,000s of chromosomes, you don't expect to see these events at all. And so, not to go into any real detail, but we're beginning to see some studies now that are reaching those sample sizes, and we're beginning to see some genes that are coming up. We don't know what's going to happen, but we're hitting the genes at the beginning of the spectrum that have the higher frequency of loss of function alleles. And it'll take moving down into the 50,000, 100,000 kind of range before we begin to hit the middle of that distribution and see what effects we may see. But there's beginning to be life in that signal, and we can bet on what will be there, but we can only tell by looking. This question of connecting variants to genes by kind of figuring out what is every regulator targeting. Well, two colleagues back at the Broad, Jesse Engreitz, who was a graduate student at the Broad and is now a Harvard Junior Fellow, and Charlie Folco, who is a graduate student in the lab, have been trying to ask, could we just figure out the regulator to gene connection? Just to put the scale of the challenge before us, it's 21,000 genes. There are several million enhancers in the genome and thousands of cell types. So doing it one at a time is not a good idea. And so they're very interested in the question of, are there just general rules? And here is a one-slide summary of what they've been finding. The unhappy possibility is that every regulator talking to its promoter involves specific biochemical compatibility. Like there's a protein at the enhancer that has to bind to something at the promoter, and it's very fussy. What they did was they took some 1,500 enhancer-promoter connection possibilities, used CRISPR inhibition to inhibit the enhancer, and then measured carefully the effect on the gene down to 10% levels, and built a data set. And based on that data set, there's not a lot of evidence of any biochemical compatibility. Inhibiting an enhancer has roughly the same effect on neighboring, inhibiting several enhancers has roughly the same order of effects on several promoters. There's no funny business as to how that's happening. The inhibition of an enhancer has similar effects up and down across promoters. Some people are fond of domains and loops, that enhancers should loop to promoters. That turns out, just empirically, to be a very poor predictor. They play a role, I'm sure, but it's not a very good predictor. Or being in the same domain is not a very good predictor. But they did come up with something that's a surprisingly good predictor, which they call activity by contact. How much activity is there at this enhancer, as measured, say, by histone 3 K27 acetylation? And how often does it come in contact? So measure enhancer's activity and the proximity to the promoter, the contact frequency, multiply those numbers. That actually turns out to be a ridiculously good proxy for the effects. That alone explains something like 70–75% of the area under the curve for that. Now, there are exceptions, but it's not a bad starting point into trying to understand the exceptions. So at least a simple model, which is surely too simple, is that enhancers just have a generic activity and their effect on the gene depends on how often they are in contact. And now we're trying to figure out where that doesn't fit. There's work I'll mention of just taking all those GWAS signals and figuring out, like, that were turned on in that cell type. Right. So that was the first periodic table, the periodic table of the genes. I want to take a few minutes and talk about this other periodic table, because it's kind of fun. I'm still dabbling around in it and trying to figure out what one can learn, and I just want to tell you about one interesting project that you might find useful for something. So this is kind of dessert after a main course. Trajectories of cellular development. So that's our topic. I don't think I have to tell anybody that we've had this revolution in single-cell biology. Not so many years ago, we couldn't analyze the gene expression pattern of single cells. We can. I'm particularly grateful to my colleague down the hall, Aviv Regev, for the work she's done to help drive this, including to help launch an international human cell atlas project, which scientists in 55 countries are involved in right now, including here. And I love talking to Aviv about this. The common thing that is done in many places is get a tissue, dissociate into single cells. Every cell's gene expression pattern, there's 20,000 or so genes, is a vector in 20,000 dimensional space. So every cell is a point in 20,000 dimensional space. A cell type is a cluster of cells in 20,000 dimensional space. And there are a lot of good tools for building these clusters and looking for subtypes and things like that. But of course, development is a trajectory in 20,000 dimensional space. When cells go from, any tissue develops, like fertilized egg to a whole organism, it starts off as one gene expression pattern, and then it moves around in 20,000 dimensional space, and it bifurcates, and it does things. Can we reconstruct these patterns? So that's the problem I'm just going to close by talking about. Reconstructing the trajectories by a method called optimal transport. This is first part is just a little bit of theory, mathematical theory with a cameo appearance by Napoleon Bonaparte. So this idea of trajectories in space is an old one. It goes back to Conrad Waddington, the great British biologist, who talked first about developmental trajectories as railroad tracks, and then later revised it to marbles rolling down the famous Waddington landscape of development. So can we get those landscapes finally? Can we get the marbles and see what they're doing? We'd want to know what cell types there are, what probabilistic trajectories. Given us a marble at one point, where did it likely come from? Where's it likely going? But the problem is, even with this super duper fancy single cell analysis, we only get snapshots. We can go to time 1 and see what cells are there. We can go to time 2 and see what cells are there. We can go to time 3, but we can't follow the same marble down the path, because every time we read out the cell, we destroy it. So we're not actually following the process. We have to somehow connect the dots. So it's as if you had a school of identical fish swimming around in 20,000 dimensional space, and you looked at them, every 2 minutes or something, and you have to figure out, like, who went where, even though they're not marked. And, it's a mathematical problem. At any time point, you have a distribution, and you imagine this is a time-varying distribution, and you have to connect up the points. And you might imagine that since I got to get one distribution turned into the next distribution on average, nearby things should go to nearby things. So it's like, suppose I had piles of dirt at time 1, and then I got piles of dirt at time 2 that it's supposed to be. I'd like to figure out, how do I move the dirt from here to there in the most efficient way with the minimal optimal transport of the dirt? So it's turning piles of probability, we'll just think concretely, are like piles of dirt, and there's a new distribution of dirt over here. So it turns out this formulation in terms of dirt is the original formulation of the problem that Gaspard Monge in the 1780s thought about, of how you move piles of dirt from one place to another place. And it was a practical problem because it was involved in how you build fortifications. If I have big piles of earth and I need to build forts, and building fortifications in Egypt with these approaches. And it was also, by the way, the origins of things like linear programming and all that. So Jeff Schiepinger, who's a really good statistician, adapted this to the moving probability piles. And the only difference is that the dirt doesn't like grow and divide and cells do, so he had to change it so it could accommodate cellular growth. But I'll leave aside the details. So then, to apply it to an experiment, we tried classic Yamanaka reprogramming. So it turns out that Yamanaka reprogramming has been analyzed a lot, but mostly by bulk sequencing, just at different time points, take all the cells and sequence them, but you can't see what's going on at the individual cell basis. It had not been analyzed at the single cell basis before. And so you only had very vague general statements. There were waves of this, and some strange genes pop up. And so it was an opportunity to collect a real data set. And so Jian Shu, another person in the lab, spent 18 days in the cold room, or something like that. Basically, every 12 hours, he collected a new sample of cells being reprogrammed by Yamanaka-style reprogramming. And he actually did all this in duplicate, and he also used 2 different media, one serum, one 2I medium. All told, Jen collected 315,000 cells across a ridiculously large number of time points. And he did generate 8 billion gene expression patterns for this experiment. So now, how do you look at 315,000 cells in 20,000 dimensional space? Well, you got to project it down somehow on 2 dimensions so you can look at it. There are things called force layout embeddings. This is a force layout embedding where 20,000 dimensional space has been projected down onto a 2-dimensional PowerPoint slide, and the points that are near each other are supposed to kind of be near each other in 20,000 dimensional space. And broadly speaking, it looks like a chicken, right? Like there's this head and a beak up there and a wing and all that. So we just refer to this as the chicken. Not all problems look like a chicken, but when we embedded it, it turns out that Yamanaka reprogramming looked like a chicken. And so you could like label the points by their times. These are points from every 12 hours across 18 days. And if you label them by time, it's actually remarkably orderly from day 0 over there, the little black thing at the tail of the chicken, all the way up to reprogrammed cells up there, or the red guys up at the top, are late cells. In fact, I'll play you the movie very quickly of the time is ticking off, and these are just where those cells have. We didn't put the cells in this place. It's just this tells you where the cells chose to be. Those are the 2 reprogramming conditions. They're now going into different directions, becoming different kinds of cell types. And there you go, day 18. You could also label the chicken with gene expression patterns for any gene you want. So if you wanted to label it with, I don't know, nanog, there on the 2nd row, 3rd column, it's obviously that pluripotency gene is up there in the head of the chicken. But things like CDKN2A, something that you might think is associated with apoptosis or something, that's more over there. You can take signatures of pathways and cell types, sets of genes that are characteristic of something, and you can find, proliferation in the 1st row, 3rd column. It's along the belly of the chicken. ER stress is up there. Neural identity is on the wing of the chicken. And so you can mark the chicken up, and for our purposes, there's at least 5 important places. There's stromal-like cells over there, neural-like cells, placental-like cells, IPS-like cells, and cells that are undergoing a mesenchymal to epithelial transition. And you can figure out what are the trajectories between these things. So I'll just give you a very quick tour of the chicken. The stromal and MET regions diverge early. Within a day or so, they're already clearly beginning to diverge. You see first that fibroblast identity goes away, and then on one of these trajectories, it returns, going up to the stromal cells. It goes away and returns, and that has lower proliferation. Instead, coming along the west coast, you'll see there, proliferation rate remains high, and the fibroblast identity never returns, and there's distinctive transcription factors. We can also see that these destinations, placenta, neural, and IPS, are not determined before day 8. that in fact they all go through the MET region, and their ancestors do not look any different before that date. After day 8, their ancestors diverge, but not before day 8. So we can immediately plot when fate determination is occurring. We can, by looking at those who choose to go on to become iPS cells, look at the transcription factors that are turned on along that pathway. And we can see known transcription factors and some new ones. I'll flag this guy OBOX6 as a previously unknown transcription factor that plays a role. We can also order this relative to events like X reactivation. There'd been a whole paper and a whole study experimentally to figure out how to order where did X reactivation occur in pluripotency programs, but you can just read it off the map of where it's occurring. The placental cells are not just generalized placental cells. We see different programs of placental cells that appear to correspond to different cell types known to exist in the placenta, different trophoblast-like cells, as well as some other primitive endoderm-like cells. The neuronal cells, same sort of thing. We can see that along the wing of the chicken is actually development from early neuronal precursors out on the tip of the wing to mature cells. We can infer genomic instability. We can actually read genomic events from gene expression, because if 25 housekeeping genes in a row are all low, it's not an accident, it's a deletion. If they're high, it's an amplification. And we can show that in the stromal regions there are clearly deletions in excess there, not surprisingly for cells that are undergoing apoptosis. And up in the trophoblast regions, we see more amplification, not surprisingly given the endoduplication that occurs in placental tissue. We can also make guesses about potential paracrine interactions. Like, for example, the stromal cells are secreting cytokines. We see clear evidence that they have genes on for all sorts of cytokines. And we can ask, at the same time that they're secreting cytokine X, is there some cell type that is expressing the receptor for cytokine X? And for example, GDF9 is being secreted by the stromal cells, and at the same time these precursors to induced pluripotent cells have the receptor for it. So we ran a couple actual lab experiments to test the computational predictions, and sure enough, that transcription factor OBOX6 that I said turns on along the path to pluripotency actually increases the reprogramming efficiency. And the cytokine GDF9 that's appearing at the same time as its receptor is appearing in precursors, it too, when you add it in a dose-dependent fashion, increases the reprogramming efficiency. So anyway, that's roughly a summary that by, again, using these kinds of information, cells in 20,000 dimensional space, and figuring out how to connect them up mathematically into trajectories, you can learn a lot. And I could say a lot more, but it will be in a paper that will be coming out. But it's an example of, again, the power of thinking in terms of these general periodic tables of cell types and signatures and things like that. So I will just end by saying some acknowledgments here. First, this first part on the genetics of common disease, part 1, phase 1 and phase 2. I wanted to put up the people I had specifically noted and acknowledge them again. And I also want to note that many more people have been involved in this, and I want to acknowledge all of them as well. And part 2, this paper that finally will appear that we have been working on these cells. And I will stop there and just say again, what a wonderful thing it is to be back in Finland. It's just, I got so much energy coming here. Thank you so much.
And I think we certainly have to say what a wonderful thing it is to have you here, opening our minds and challenging us with such exciting things. So thank you, Eric. Do we have time for just a few questions, if they are urgent and exciting? Otherwise, we can
go have tea.
Ask in coffee.
Yes, exactly.
Coffee, okay.
Surely someone will overcome their Finnish reserve.
No, that's
yes,
not
well done.
Juha.
Okay, I knew I could count on Juha. You have a microphone if you want to.
Okay, thanks.
It's right there.
Hello, Eric. You started by saying that you are from New York and your David Boston is from other part of New York.
Bronx. Yeah, I'm from
Right.
Brooklyn. He's from the Bronx.
The
Yeah.
Bronx. So my godmother used to live in White Plains, New York. So I may qualify as a contestant for discussion in this respect, if you don't mind.
That's close enough.
Okay,
Yeah.
So the only thing that, of course, we have this Finjin thing and lots of things happening now in Finland.
Yeah.
And I've read carefully the papers about these polygenic scores and so forth.
Yes.
Now I have a problem with all this.
Good.
Okay. And I like to go back to old things as you do. And
Yes.
and so one of those is the good old twin studies
Yes.
that tell us that kind of the genetic component or the heritability, or let's say simply the concordance ratio in monozygotic twins
Is not one.
for coronary artery disease is 40%.
Yeah.
And so the thing is that, and we understand why that is that way. So it's, of course, the process of atherosclerosis depends on genes and food
Environment.
and lots of things. But then there is the random component of getting any one of the plaques inflamed and then rupturing and then actually causing the phenotype that we care about.
Yep.
So my problem is that what is the utility of doing the risk score when the advice to the low-risk individuals is exactly the same as the high-risk individuals?
Oh. Is that always the case? So they just recommended mammography. The age for mammography should be increased to 50 in the United States from 40. Is that really correct? We're deploying medical resources that we can't give to everybody that are not without risk in some cases. So in many of these, we can think about heart disease. Yes, everybody should have a healthy lifestyle. Everybody should exercise. Probably Joe Goldstein would tell you everybody should just take statins. We should put them in the water. Sure. But, even then, not everybody listens. So it might be that people who have higher polygenic risk scores should get more of the social support attention to actually do those things that we all should do but don't do.
Do we know that they would care?
We don't know. We don't know. But we do know that if we were going to deploy resources, we would want to deploy them where they might have the biggest benefit. Now, do we know how to get people to comply or do these things? Not necessarily, but we might want to learn. With mammography, I think we really do want to devote resources in the right way. So in no sense do I want to say any of this is simple. Figuring out how to deploy resources, finite resources, in a medical system for people who are busy doing lots of other things. Each disease will be different. It may be in some cases that we're talking about an intervention that has no risk at all and everybody should just do it. But in other cases, there may be risks and benefits to be balanced. We may have a lot to learn how to get people to do things. And these are hardly perfect predictors. And we have to be honest and say those things. On the other hand, for the first time, we're beginning to get things that actually have a serious population health benefit, and we don't know how to deliver them. But we do know that if we did deliver them, they could make a real difference. So for those reasons, never trying to be, never wanting to overstate it, I also don't want to understate that this should become an important part of medicine. And we have to figure out how to make it an important part of medicine, and it's not going to be easy, but the best place to do it, in my opinion, is Finland.
Yes.
Do I?
Excellent. All right.
May I
Thank
still-
you, Eric. Yes,
We can talk more over a coffee.
let's have coffee now. Okay.
Let's
Keith.
go have coffee.
All right, yes.
Thank you.
All right, I hope I got a call.